UCSF with Wendell Lin, where he examined the molecular and evolutionary logic of cellular decision-making through reconstitution of the budding yeast MAP kinase signaling network. After graduate school, he took a break from academics and founded a startup company called Cell Design Labs. It's an immunotherapy startup company, which, as I understand it, engineered drug-responsive chimeric antigen receptors. Yes, sounds very cool. And it was recently acquired by Gilead about 14 months ago. So he then went back to academics and started a postdoc at Stanford in Manu Prakash's lab, where he's been studying how mechanical behaviors are molecularly specified and programmed in ciliates. His talk today is titled Deconstructing Cells to Understand and Engineer Microscale Molecular Machines.
All right, well, thank you so much for that introduction and the opportunity to be here to tell you about the work that I've been doing, which is really about cellular machines, which are some of the most sophisticated molecular machines in existence. Cells are capable of organizing hundreds of millions of proteins across hundreds of thousands of different interactions in the cell to perform complex functions and specify sophisticated behaviors. And many of the things that cells can do are really go beyond anything we understand how to do with molecules ourselves. They can investigate, analyze, and respond to objects in their environment, like this T-cell interrogating a target, or this unicellular predator recognizing its prey. They can organize and coordinate molecular processes across diverse length and time scales, like a 24-hour cell division, or a minute's timescale calcium oscillation. And they can even manufacture other molecular devices that can function independent of the cell, like motile cilia that can be used for propulsion, or anucleate delivery systems like platelets. And so when I see a cell, I don't just see interesting biology to understand, but I see a window into the design principles of sophisticated molecular machines that operate at the microscale. And while we know a lot about what cells are in terms of their molecular composition and spatial organization, we know far less about how complex functions actually emerge from this macromolecular hardware. And it's a challenging question because the fundamental building blocks are very small and operate on very fast timescales, whereas the machines that cells build are micron-scale devices that perform their functions over minutes, hours, or even days. And this mapping between molecules and cellular machines is a fundamental one at the intersection of biochemistry, systems biology, and cell biology that's really fascinated me throughout my career. And I want to tell you about different approaches I've taken to try to understand this mapping. I'm going to tell you a little bit about previous bottom-up approaches in which I reconstituted signaling networks in vitro to ask, how can cells use molecules to build circuits that organize information or control molecular assembly processes? And then I'll tell you about what I'm currently doing of taking a top-down approach to ask how animal-like behaviors in unicellular protozoa can be understood as the outputs of cellular machines that are programmed largely through signaling and mechanics working together. And then I'll briefly close out by telling you what I'm thinking about doing going forward, which is to integrate these approaches together to continue understanding as well as building new classes of molecular machines at these length scales. And so I'll start by telling you about these bottom-up approaches, which are really about trying to understand how cells can use molecules to achieve control. And control is a really critical aspect of how cells are able to execute specific functions like we see here. And when loss of control is often associated with entering into disease states. And the way in which cells achieve control are through networks of signaling molecules that can sense things that are going on around them, process that information within this network, and then direct specific responses that change and alter their behavior. And when we talk about signals propagating or being processed in these networks, there's really two key mechanisms at play. There's enzymatic cascades, like when one kinase phosphorylates another, and there are molecular assembly processes, like when an activated GTPase recruits an effector to the plasma membrane. Now, because networks are made up of multiple of these activities working together, I've taken an approach of reconstituting multiple components into circuits to try to probe their higher order systems level functionality. And this is analogous to how you might try to take electrical components and explore the space of circuitry available to them on breadboards. And so I want to tell you today about how this network reconstitution approach has given us insights into each of these core signal propagation mechanisms, starting first with enzymatic cascades of kinase phosphorylation. Now, something you often see in signaling networks that's confusing are branch point architectures like you see here, where it's not immediately obvious how a cell is going to be able to control which way signals propagate. And a particular branch point architecture I studied in great detail can be found in budding yeast, where there's a kinase called ST7, which is activated by either starvation or pheromone inputs, But when a starvation signal is applied, signals will flow to this KSS1 foraging kinase, whereas when pheromone is applied, signals will flow to a mating kinase called FUS3. And a clue to how the cell might be controlling which way signals go came from genetic evidence that had identified a scaffold protein called STEAP5, which selectively associated with components of the mating pathway. And so to try to understand how this branch circuit might be functioning, we reconstituted a mini yeast map kinase signaling network made up of the various kinases, scaffold proteins, or input-specific signals like lipids, which they could then reassemble in vitro reactions and query the resulting circuit behaviors under a variety of different conditions. And one of the first things we discovered with this approach was that signals could propagate to the KSS1 kinase independent of the scaffold protein, whereas signals could only propagate to FUS3 if this scaffold protein STEAP5 was included. And it turned out what was happening here was that the scaffold protein was allosterically regulating FUS3, altering its kinase structure to make it a better substrate for phosphorylation. Now it turned out that this scaffold activity itself was also subject to regulation because we could identify two distinct activity states of the scaffold in our reactions. An inhibited state in which signal transmission was inefficient, and an uninhibited state in which signal transmission was efficient. And by including PIP2-containing liposomes in our system, which mimicked the effect of pheromone stimulation, we could actually drive the scaffold into an active state, suggesting that mating inputs directly activate the scaffold protein. And what this means is this combination of scaffold dependencies and scaffold regulation make it very easy for the cell to control which way information flows inside this network. Because if a starvation input is applied, the scaffold protein is inactive, and signals will flow to this KSS1 kinase by default. Whereas if a pheromone input is applied, the scaffold protein activates, and now signals are permitted to flow to FUS3. Now, what I just told you is a rather complicated allosteric control scheme that makes this circuit here work. And if you're wondering why the cell didn't just build two separate signaling pathways instead and forego all of this, well, one reason is that pathways like this are built through evolutionary processes of duplication and divergence. And when I looked across the fungal tree of life, I could actually identify intermediates on this pathway that contained either a single kinase or duplicated kinases that have redundant function. And within these genomes, there were no STEAP5 scaffolds present, so we can think of these as pre-STEAP5 signaling networks. And something I realized was that the same reconstitution approach we were using to understand how this budding yeast circuit worked could also be used to give us insights into the evolution of these particular control mechanisms. And the idea was that we could purify a variety of kinase components from across the fungal tree of life, and then use them to reconstitute synthetic circuits that had never built from components that had never interacted with one another during the course of evolution. And in particular, we could start to ask questions like what effect a scaffold protein might have on pre-STEAP5 mechanisms. I just want to share one result from this evolutionary study that I thought was very interesting, which was that we found that many of these pre-STEAP5 kinases could actually be allosterically regulated by the STEAP5 scaffold. And this was really surprising to us. But not only could some of them be activated, within a pool of otherwise functionally equivalent kinases, within this pool of otherwise functionally equivalent kinases, some were more FUS3-like with respect to this property, whereas others were more KSS1-like. And what we think this means is that these types of allosteric mechanisms that seem very confusing to us may actually end up being very natural ways with which a cell could discriminate duplicated components from one another that are otherwise very similar in terms of their overall structure or activity. And thus, this particular scheme that we see in budding yeast is just an example of this strategy following evolutionary refinement. And these use of allosteric mechanisms to discriminate branch point architectures may be a general solution to resolving these types of problems. But remember that enzymatic cascades are just one mechanism by which signals propagate in these networks, and another is through organizing molecular assembly. And I think this is really best exemplified by the small GTPases, RAS, which are signaling powerhouses inside eukaryotic cells. They can respond to many different signaling inputs and can communicate to over 60 different downstream effectors, enabling them to produce a diverse repertoire of very complex signaling outputs. And for those who don't remember, the way that RAS signaling works is it can exist in an off state in which it's bound to GDP, and an on state in which it's bound to GTP and can interact with downstream effectors. Now, RAS can move through this cycle on its own, but this intrinsic cycling is very slow. And so the cell has enzymes called guanine exchange factors, or GEFs, that can turn RAS on, and when GTPase activating proteins, or GAPs, that can turn RAS off. And each of these individual proteins and steps within the RAS catalytic cycle have been studied